Oh, good.
Um, so, um, Perfect. We're recording and we can get started. So, thank you again, everybody, for joining us today. We're going to be continuing our series of lectures around pluripotent stem cells and the pluripotent stem cell workflow. Today's lecture, we're going to be talking about stem cell differentiation. We actually have a distinguished guest, Dr. April. Pyle is here with us to also talk about some of her work around mesoderm formation and skeletal muscle formation. But before we get into that, I wanted to just give you, again, a brief introduction into differentiation in general. So, for those of you who've been following along, we have been talking over the past several lectures around about pluripotent stem cells, right? How to generate them via either isolation or reprogramming, how to culture them in 2D and in 3D. Um, uh, Tom last week also talked about how we can use new genome editing tools to edit the genome and make specific edits into our cells before we differentiate them. And today we're going to be talking about that final process of these cells transitioning out of that pluripotent state and into and going down a differentiation path to eventually give rise to all the different cell types in your body, over 200 different cell types. So before I do this, I actually found this video online, and I wanted to share it with you just so that we can just to kind of set the stage and to show you this process, make it maybe a little bit more real. So this is a video from a fertility clinic in the UK showing the development of an embryo. This is a day three embryo. As you can see right here, the cells are dividing, right? And what I want you to pay close attention to, I think there's somebody on, if you could mute yourself, perfect. I want you to pay attention to is as these cells are dividing, they're changing in morphology. Right now at the morula stage, you're seeing the blastomeres compact, right? And eventually, as these cells continue to divide, their position within the embryo becomes very important. As you can see now, these cells start to shift, and actually, we have the first break in polarity where we now have at the blastocyst stage two different cell types, right? We talked about this. The outer cells are your trophectoderm, which will give rise to your placenta, and then this here is your inner cell mass, which constitutes the population of pluripotent stem cells that we usually talk about. Um, so just keep this in mind as you're going forward, right? This is a really, really cool process where you can see firsthand how at an embryo level these cells are changing, moving around, and actually adopting different fates, right? And so this process is really, really important to kind of understand as you're starting to think about differentiation, because as we mentioned before, pluripotent stem cells. See if I can get my pointer going here. Pluripotent stem cells have the capacity, by definition, to give rise to all cells within the embryo proper. So they can specify ectoderm, mesoderm, and endoderm lineages, right? But some of you may be wondering, well, how does that decision happen, right? How does a pluripotent stem cell decide to become a cardiac cell, or heart cell, or a skin cell, or a neuron, right? And in very particular, and a very particular organizational scheme, right? For the most part, our bodies have an axis polarity. We have, a, a, a generalized body type that we look for to what we call normal, right? So how do these cells know where to go and what to differentiate into? Well, this process is governed by a bunch of different factors that have some of which I've outlined here, right? The paracrine and autocrine signals from within the cell or the surrounding cells, the signaling that happens there, of secreted molecules will help determine which how these pluripotent stem cells decide whether to go and become mesoderm, endoderm, or ectoderm. There's also maternal cues and hormones that govern this process, right? Within from the mother. The other factors include things like physical pressures and forces, right? Cell-cell contact. We talked a little bit about this, how stem cells like that tight stem cell contact, right? But as you know, as these cells start to move and differentiate, they're exposed to different cell types, different potentially different environments, and that is really, really important, right? That dictates the outside environment for the cell, right? What it's in contact with. So it does affect the signaling within the cell itself. And then finally, we also have things like oxygen tension or nutrient availability that are also really, really important that dictate this process of differentiation. And typically, we'll touch on this again. What we try to do in the lab when we differentiate our pluripotent stem cells into a specific cell type is we try to mimic these signaling pathways that are or cascades that start in a particular towards a particular lineage. And we'll talk a little bit more about this. So as you're thinking about that, right, all these things that govern this differentiation process, I also want to take a little bit of time to just appreciate how insanely complicated this process is, right? All these factors that we just talked about really are a way of the for pluripotent stem cells or cells in general to sense their outside environment and then react to it, right? So the signaling pathways from the cell surface to the nucleus that facilitate differential gene expression that can then specify either mesoderm, endoderm, or ectoderm are governed by a bunch of different cell signaling pathways. I don't want to date myself, but when I went to first several years in college, we had 7 pathways that we knew about that we studied and we had to memorize. Right now, I think that list is 14 plus. But I've just put a couple of those pathways on the screen and that you can see here. Wnt signaling, BMP signaling, MAPK, VEGF, FGF, AKT, right, signaling. And you'll notice that at the cell surface, right, these proteins or receptors interact with a bunch of different effector proteins, some of which converge, right? So these pathways actually also crosstalk to each other. And altogether, what you end up doing is sensing the outside environment and reacting to it internally. And this is a map of a metabolic map of all the different metabolic activities that go on in one cell, right? So these signaling pathways converge onto the gene expression side to allow a specific protein production that can then produce these proteins that outline a metabolic pathway for a cell and then govern its function eventually, right? So beta cells, for example, are their metabolic or gene expression pathways allow them to secrete insulin, right? Very different to cardiac cells, where their functional output would be to contract in order for your heart to pump blood to the rest of your body. So really, really cool process, but super, super complicated, right? And specifically for today, we're going to be talking about mesoderm specific lineage formation, right? So if I take you back to the image of that blastocyst, right, that we saw, I mentioned that at this stage, we typically have 2 different cell types, your trophectoderm, that are these outside cells, and then your inner cell mass, which are the cells that are going to give rise to your embryo proper, or all the cell types within your body. Now, as the embryo implants into the uterine wall and it starts to invade into the maternal bed, the inner cell mass here actually further specifies into 2 different subgroups of cells. The hypoblast, which is the cells that are in the darker color at the bottom here, and the epiblast as well. The hypoblast will eventually give rise to your endoderm lineage, while the epiblast will give rise to the ectoderm and mesoderm lineages, right? This is quite a bit involved. I know you don't have to memorize or know this in great detail for now, at least, right? This is basics developmental biology. But what I want you to know is during this step, right, as the embryo starts to gastrulate, this is when, the three germ layers are starting to be specified. These two layers, the epiblast and the hypoblast that are pictured here, start to migrate one towards the other. So the epiblast on top starts to migrate and pinch inwards to create the neural tube. And in this process, you actually end up also specifying the mesoderm, which by definition, right, meso is the Latin word for middle. Mesoderm is basically the layer of cells that forms in the middle of the epiblast, so right here. And mesoderm is actually divided into three different layers, if you will. There is the paraxial mesoderm that's closest to the neural tube that will eventually give rise to skeletal muscle, vertebral joint, and cartilage, as well as skin. There is the intermediate mesoderm that gives rise to your kidneys and your gonads. And then there's the lateral plate mesoderm, which will give rise to cell types like, cardiovascular cells, like your heart and blood cells, blood vessels, blood cells, as well as connective tissue and limbs, right? So um, for the rest of this particular part, we'll talk a little bit more about some of those cell types. I'll touch upon a bunch of cell types on the cardiovascular side and the blood side. And again, April here, do you want me to continue on with the cardiac side, or should we?
Sure, you can do cardiac and then I can jump in with skeletal muscle.
Let's do that. So on the cardiac side, right, if the goal of your research, right, in vitro is to then, after understanding this particular process, is what we want to do in the lab is mimic that, right, in an in vitro setting where we can take pluripotent stem cells and then trying to modulate the different pathways that govern the way that they differentiate specifically into epiblast, mesoderm, and then specifically in our case for cardiomyocytes, lateral plate mesoderm, right? The idea is that you want to go through these progenitor stages in order to get to your final terminal cell type. And we know from a lot of developmental studies that this process is governed by a bunch of different signaling pathways, including Wnt signaling, FGF signaling, BMP, and sonic hedgehog specifically. So what we do in the lab is, in pretty much in any differentiation protocol that you'll see, is we're trying to expose our pluripotent stem cells to a particular growth factor that will signal to them that they should start a particular cascade of events that will eventually lead to them, to them differentiating into a particular progenitor state, and then that progenitor state can then be further pushed into our terminal cell type, right? In the case of the cardiomyocytes, we take our PSCs into mesoderm progenitors, right? And then cardiac mesoderm before we specify cardiac progenitors that will eventually mature into cardiomyocytes. And this work has, there's a lot of folks that have been working on this and for quite some time. So there's several differentiation protocols that are widely used and sort of accepted in the field. This is just an example of some of those different differentiation protocols. And you'll notice here, and some of you may ask this question, say, well, how does that make sense, right? Why do you have different differentiation protocols to derive the same cell types, right? Shouldn't they all be the same? Well, the answer to that is sort of yes and no, right? Because I showed you the reason why I showed you all these signaling pathways and the metabolic, you know, disaster that is that image is just to show how complex that our cells are, right? And so what we try to do with these differentiation protocols is make alterations to mimic the signaling pathways, but that can be done multiple different ways, right? And so you might see different differentiation protocols for the same cell types throughout the literature. This is not just true for cardiomyocytes, but could be true for other cell types as well. And you'll often see that, folks in our field will have heated debates on what they think would be a better differentiation protocol that better mimics this developmental pathway, right? But again, I digress. These are just some of the examples here. So around 25 years or so now, Gibstein published a protocol on how to take PSCs and through the formation of embryoid body, differentiate them into beating cardiomyocytes using FBS. This was a little bit of an older system, a little bit hard in terms of generating or efficiency to generate these cells. So folks continued to develop on that process, and in 2010, Gordon Keller published a protocol that uses growth factors specifically in order to try to mimic that signaling pathway, right? BMP, FGF, activin, VEGF, and Dickoff are some of those growth factors and cytokines that are used in order to get these cells, again, to generate mesoderm, then cardiac mesoderm, and eventually have beating cardiomyocytes. In 2012 and close to that, in around 2014 as well, Sean Pelichek in Wisconsin, Joseph Fu at UCSF also came out with protocols where instead of using growth factors, they were using small molecules in order to also do the pretty much the same thing, right? Is to in this case, shear is a Wnt activator or Wnt agonist. It's a small molecule that targets GS3K beta, part of the canonical Wnt signaling pathway, and can start the induction of mesoderm. And then shortly after that, you need to actually inhibit Wnt signaling right after that. So they add another small molecule called IWP2 that blocks Wnt activity. Which will then take the mesoderm progenitors and start to specify them into cardiac mesoderm specifically. And then the last option in terms of differentiation protocols here is there's also some commercially available kits. At Thermo, we have a particular kit called the PSC cardiomyocyte differentiation kit. That's ready-to-use kit that comes with two different buffers, buffer A and buffer B, that again do the same thing here, right? And so we sequentially change media from buffer A to buffer B that coincides with that transition from mesoderm into cardiac progenitors and eventually into a cardiac maintenance media that helps continue to mature these cells into cardiomyocytes. Any questions so far? I know that that was a lot.
Yeah. Hi, Omar. I have one quick question. So
Yes.
I have actually two questions. The first
Yes,
is
go
like,
ahead.
yeah, right now I'm using your PSC cardiomyocyte differentiation kit, and this is like I would like to endorse this one because this is a very good kit I have used so far. And right now, actually, I have one culture that is going on. Maybe expecting beating in the next week. Yeah, pretty much good kit this is. Apart from this one, I wanted to get some information. Like whenever we present the data, they are asking like, okay, so how you have differentiated? Then it looks like I am telling that okay, I'm using buffer A, then buffer B, but I do not have any clue. I do not want to know like all the composition what you have, but whether this is based on Wnt inhibition or like any growth factor based. So if you have something that you could share, like what is the basis of this differentiation, that would be good thing information. And my second question is you have shown different method for differentiation of the cardiomyocyte. So after using this protocol, like what could be the yield of cardiomyocyte, about the purity and contraction?
Good
Yeah,
questions.
thanks.
Yeah. So I'll answer your second question first. So in terms of the different differentiation protocols, you're right. They can potentially specify different different subsets of cells. And And this is why I mentioned that different researchers will actually, prefer one particular protocol over another, depending on what they're studying per se, right? So this can be a little bit challenging to answer. There isn't a cookie, I don't know why, what's the expression there? Cookie box cutter? No.
Cookie
Cutter
cutter.
Cookie cutter. Thank you very much. A cookie cutter answer there. But we'll talk a little bit more about this when we mention the 3D differentiation part as well. Is um, we are limited in the scope of a lot of these protocols that you see here, for example, are in two dimensions, right? And we're not two-dimensional beings. We're three dimensions. So that cell-cell contact and 3D aspect might be important for a particular function of those cardiomyocytes that won't be conferred with a 2D protocol, for example. So there is benefits to different protocols, but um, there doesn't necessarily mean that one is always better than the other. Does that make sense? So it
Yeah.
really has to be an evaluation of the phenotype of the cells that you are deriving at the end. Typically, with cardiomyocytes, we look at a bunch of different markers, right? Troponin, alpha actinin. We look for beating, and then we can also look at beating rates, rhythms, rhythmic voltic changes. Some of which we'll actually look up here as well, or look review together in the next several slides, Naresh. So don't worry. In terms of the first question that you asked of what's in the cardiomyocyte differentiation kit, this is a little bit more nuanced of an answer, right? And I'll give it maybe not just for thermo, but in general, generally speaking, commercially available kits tend not to, advertise too much of what's inside of them, just for the intellectual property, kind of the commercial aspect of it. So whatever information we can give to you, we will. Let me know. You can email me. I know we've emailed before. And I'll try to see if there's specifics that we can answer around what you want to know as part of that kit as well.
Yeah. Okay, good. Thank you.
Of course. Okay. So um, Naresh mentioned the cardiomyocyte differentiation kit, right? So our protocol that we have is we typically take cultures that we grow in StemFlex in our StemFlex medium. We grow them to a particular confluency, and then on day zero we can start our differentiation, right? Where we add media A that's going to drive mesoderm formation, right? And then 2 days later, we switch the media to medium B, and that should start to drive cardiac specific differentiation, right? And once you do that, let me see if this will work, hopefully. So in about 9 to 10 days, you should start to see these cells contracting, right? Which is the function of what your cardiac cells, or what the function of cardiac cells is, right? These cells are basically, they contract in order to pump blood to the rest of your body. And so you can see this like wave pattern. These cells are basically all contracting in a sheet, which makes this wave pattern because one of them is pulling, and then it kind of does that little wave motion. But we can also look at some of the intracellular signaling that we know happens in these cardiomyocyte cells in general, mainly this influx of calcium into the cell. And we can track that with different dyes. Like this is just an example of one of those dyes, a fluorophore. And you could see how we can have these firing bursts that coincide with when these cells are contracting, right? So we can start to analyze how these cells function and relate that, to us deriving them from our pluripotent stem cell populations. Um, with that being said, right, I just mentioned not too long ago that most of the methods that have been developed on the differentiation side have been in two dimensions, right? On a monolayer, which has served as a really, really, really useful tool and continues to do so. I strongly, strongly believe that. But there's a lot more work that's starting to look at how we can continue to expand on our understanding of this 2D models and perhaps make them a little bit more complex. where we can start to incorporate the 3D aspect of, or the 3D biology aspect of, into the differentiation process, right? And this is true because, if we want to get a good understanding of what happens in vivo, we really have to recapitulate this process more truthfully, right? So when we're studying the heart, we want to, be able to look at different cell types, all at the same time, if for certain biological questions that we have to ask, right? Not for everything, but we want to potentially even look at that cell-cell contact in that 3D context, right? And whether that makes a difference to the way these cells function, the way these cells behave. And given the fact that we're using a pluripotent stem cell derived source, can we use this can we leverage this in terms of an expansion capability, right? If we need to address some sort of disease or pathology with some sort of cell therapy, right? A cardiomyocyte cell therapy, we have ways in which we can grow pluripotent stem cells and scale them up quite readily. And if we have an indefinite source of these cells, could we then also technically have an indefinite source of our terminal cell type, right? That we can use for some sort of therapeutic application. So um, it's really important to note here that a lot of the differentiation protocols that you'll see will either play in one of those two spaces, right? They'll be 2D systems or monolayer adherent systems, or 3D or slightly more complex or organoid systems, right? And there is a little bit of a gray area between co-cultures as well, but typically what folks are seeing is, or at least claiming, is that 3D systems and models tend to be more physiologically relevant, right? That they can potentially recapitulate certain aspects of developmental biology in vitro as well. And then that uh, uh, this could potentially be a better approximation of what really happens in vivo compared to 2D models. This isn't always true. I really have to preface that. It's very dependent on the cell type, on the differentiation protocol, and on the biological question that you're asking, right? Um, these very complex 3D systems can give you a lot of information, but it can also give it to you with a lot of noise in the back that is very hard to parse through what's true or what's not, right? A lot of variability. So you really have to make sure that you pick whatever system it works best for whatever question that you're asking. For us, we for the 3D differentiation of cardiomyocytes specifically, we have already mentioned to you guys how we've used one of our PSC media systems, right? Our stem scale PSC suspension medium to culture our pluripotent stem cells as spheroids, right? That you see down here. Let me see if I can get my pen one more time. There we are. You see down here. So these are pluripotent stem cells that haven't differentiated yet, right? These are just grown as PSC spheroids, and we can grow them in these really nice cultures. We can maintain their size pretty well. And so what we've done at Thermo is we've used those pluripotent stem cell spheroids that you see in panel A that were cultured in our stem scale medium, and we use those cells as our starting point for our differentiation in 3D. And we tried several different differentiation protocols, and I'll show some of that data in just a bit. But where we can start differentiation into mesoderm and eventually cardiac mesoderm, and in about 2 weeks we can specify these really nice looking what we're calling cardioids or cardiac organoids that actually end up beating. And I think I may have showed you this before on our previous call. But um, uh, to give you a little bit more color here on this process, we actually worked with the Lee Lab at Harvard University in order to standardize the small molecule differentiation protocol and kind of adapt it here, if you will, where we can take our pluripotent stem cells that are cultured as spheroids, again in our stem scale media, and then use the Wnt agonist here, right, to activate Wnt signaling for the first day or two in RPMI media plus B27, making sure that the B27 is a version without insulin. This is very, very important, and because insulin can actually inhibit mesoderm formation initially. So you'll see the first part of this protocol where we're specifying mesoderm, we have no insulin in here. That usually is one of the components that's part of the B27 supplement. After the first couple of days, we actually similar to what I said before, we use another small molecule to inhibit Wnt activity. So after we initially specify mesoderm, we want to start to specify or further differentiate these cells, and we need to stop Wnt signaling at that point, and that's why we add this Wnt inhibitor. And by day 6, we remove, we no longer have any small molecules in the culture, and we actually now re-add that insulin that was missing before, right? So we add a B27 with insulin from day 6 to day 14, and the goal here is to specify cardiac cells, which do need insulin in order to properly mature, right? And eventually, in those 2 weeks, oops, what you'll see is, and I have to keep jumping from laser pointer to stuff, but you can see these really cool organoids that specify in beat, right? I keep saying this, that this is not an AI generated image, that this is an actual organoid that we took a video of in the lab that looked like a cell with a heart inside, right? So it was very picture worthy or photo op worthy. And you'll see here as we differentiate these cells, right, that we start to see those spheroids change in morphology. We start to see expression markers that are associated with cardiomyocytes pop up or start to be expressed, like TNNT2. But you'll notice that this process is very dependent on the concentration and duration of those particular small molecule activators or inhibitors. So again, that process of fine-tuning that signaling pathway needed to generate your cell type is really, really important. I won't go into too much detail on how am I doing for time. Okay, not too bad. I won't go into too much detail here, but there's a couple of things that we learned along the way in this 3D differentiation process that I want to bring to your attention. The first is that the starting spheroid size matters in the case of cardiac differentiation. And so what do I mean by that? Right? I mentioned to you that before we start differentiating these cells, we culture them as pluripotent stem cell spheroids, like you see over here on the far left, right? And we can control the size of these spheroids by either manipulating the speed at which they're cultured or the duration of the culture period, right? So if we just leave them longer, they grow a little bit more. So we took 3 different sizes of spheroids, stem cell spheroids, and then started to differentiate them into cardiomyocytes with the protocol that I just shared with you. And as you can see, if we take these really small pluripotent stem cell spheroids and we differentiate them, they mostly die. You don't really get too much from that. If they're slightly bigger, we can specify them a little bit better. We do have some spheroids that start to beat at around day 10. But if they're around 300 microns, which again is the biggest size that we've tested here, they tend to, again, even differentiate slightly even better, where we do see evidence of those larger spheroids maturing and displaying a beating phenotype as early as day 8, right? I'm sure if you keep going larger and larger, that benefit is going to diminish back up again, right? So there's definitely a sweet spot, but I wanted you to know this because it's important to take a look at these things, right? Even though that the cells themselves within these cultures are phenotypically the same, right? They're pluripotent. We talked about this earlier. They express markers of pluripotency. They are typically karyotypically normal. You'll notice that the size of the spheroid confers that 3D biology is different here, right? That the center of these larger spheroids experiences different environments than potentially these smaller spheroids that are maybe a little bit more accessible in terms of nutrient or oxygen in the center. And this pattern that we saw with, with differentiating the bigger spheroids or the smaller spheroids also carries through if you look at gene expression. So the relative gene expression, when we're looking at certain genes that are known to be associated with cardiomyocytes, we tend to see much higher induction of those genes if we start from a larger a spheroid compared to a smaller one. Now, the other um, factor that I briefly mentioned to you was that this particular protocol is that's really important to consider about this protocol is the duration as well as the concentration of those small molecules that you're adding, right? So within the context that we were just talking about, right, the day two spheroids that are smaller, the day three that are kind of medium size, or the day four spheroids that are largely bigger, once we start differentiating them, we can take and test different concentrations and durations of those small molecules. This is an example where we tested two different conditions of this Wnt agonist, the shear that's added on day one, right? One condition was we kept it at, the recommended concentration, but exposed the cells to it for 48 hours. Another condition was that we doubled the concentration, but exposed for half the period of time, so mainly 24 hours. And we did these two conditions. We tested that with the small PSC spheroids, the medium-sized ones, and then the larger-sized ones. And as you can see, oops, in terms of a beating phenotype, Naresh, maybe this is to your point as well. The different conditions, have different effects on the outcome of our differentiation protocol. Where for us, we saw that the best condition here is, again, the differentiation of those larger PSC spheroids in a maybe slightly more concentrated, shorter duration of this Wnt agonist. And eventually, what these cells look like is, they have that really nice beating phenotype. Finally, I mentioned to you that the original differentiation protocols used basal media like RPMI that was supplemented with the B27 supplement, initially without insulin and then with insulin. But our team also wanted to ask whether a basal media that was specifically designed for cardiomyocytes would be more appropriate in this particular case. Keeping the small molecules the same and pretty much everything else the same, but switching out the RPMI and the B27 to media that was specifically designed for cardiomyocytes, which was already part of our cardiomyocyte differentiation kit. We have a cardiomyocyte maintenance media. So we used that media instead of the RPMI. And again, here, the basal media in itself did make a difference, where we can see that with the RPMI condition or test, that we needed a particular concentration of shear to start to see a beating phenotype. But in a cardiomyocyte maintenance media protocol or setting, we could get that same beating phenotype at a slightly lower concentrations of this Wnt agonist or shear, all basically culminating into a final concentration and a standardized protocol that we have. And so the reason why I'm mentioning all of this, right, is that I just wanted you to appreciate that this is a complex process, that a lot of work goes into this, and there is a lot of different moving parts that we have to understand in order to faithfully replicate that developmental, shift or shifting stages, if you will, in order to reach our final cell type. And to this end, right, when we get to that final cell type, that expresses all the right markers that we know these cells should, right? And for our protocol that I just shared, we actually can even track this across the different developmental stages, where we know that some of these developmental stages have specific markers, like, brachyury or T is a big marker of mesoderm that we always look at. Folk1 and nestin would be for cardiogenic progenitors. And then the more mature cardiomyocytes express alpha-actin and troponin. And we can follow that expression profile across development or mimic that as well. And then the really cool thing is that we can keep these cells or these organoids, these cardiac organoids, in culture for a long period of time. So we've actually kept these cultures for a little bit over 6 weeks, and then we stain them to look at the cells inside, right? We wanted to know what are the cell types that are comprising this cardiac organoid, and so we actually used a high content imaging system here. I won't go into the details of the system here, but basically because they're 3D structures, we needed to take Z stacks or look at individual planes one at a time across the entirety of the entire length of the organoid. And then we basically recomposite those into or re-rendered them into a maximal projection image, which basically tells us what cells are inside and where they are, basically based on our staining. But if you look here, not surprisingly, we see a population of cardiomyocytes that are actin and cardiac troponin positive. They tend to sit closer to the cortex, the center, excuse me, of the organoid, but they can have some peripheral attachment to invasion. But cardiomyocytes are not the only cell types that we're specifying here. In this 3D context, similarly to what happens during development, all these cues really dictate how these cells develop. So we also see some maybe potentially more connective tissue in vimentin positive fibroblast cells that seem to encapsulate the organoid across. And then we also have a layer of endothelial cells that forms from what we gather in an attempt to try to start to form blood vessels or start to start that process, right? Where we can see them outline or surround the cardiomyocytes themselves as well. So altogether, really, really cool way to look at beating, cardiac organoids as a model to study, potentially how we can administer different drugs to simulate different beat rates or fix different pathologies or diseases or disease model. And to this end, right, we've done a little bit of work where we can show that we can put these organoids through some functional tests as well, where we can take that cardiac organoid and dissociate it into 2D and actually plate it back down onto these multi-electrode assays. You can see these beating cardioids, right? And we can measure their rhythmic beat rates. Our R&D team actually went one step further, and they were able to get these intact cardiac organoids, I should say, in their 3D format, and place them on these multi-electrode assays to measure their rhythmic voltage pulses, but in a 3D context without dissociating them. In this case, you can see this little guy, we can detect a stable beat rate of 0.3 seconds. But we've also used some more sophisticated instrumentation to look at depolarization and T-wave patterns, right? Where we can see how these cells polarize and depolarize and repolarize as well. Again, giving us a really, really cool model to study different cardiac functions or pathologies as well. And so with that, I'll stop there. But just as conclusion before I pass it on to April, is that we can take these cells and either from 2D or 3D and differentiate them into cardiomyocytes specifically here. We do see some advantages that are conferred by some 3D models in terms of our ability to generate more complex models per se. but in a sense where we have multiple different cell types within that same organoid that we can study. But like I said before, 2D models are not worse if in that context, right? They have their own specific intended uses, if you will. And with that, I'll get off my soapbox for just a little bit. And April, maybe I'll pass it to you to take us through some of your work, talk about some skeletal muscle.
Sure.
Thank you.
Sounds good. You can answer questions while I share, I guess. Let's see here.
Yeah, before I just introduce Dr. April Pyle, it is my pleasure to introduce today's invite speaker. And Dr. April Pyle, she is a professor in the Department of Microbiology, Immunology, and Molecular Genetics in UCLA. And Dr. Files is the leading expert in the stem cell biology, gene therapy, especially focusing on the developing the new treatment for muscular dystrophy using pluripotent stem cell and genome editing. So she is also the co-founder of Myogene Bio, a company translating this discovery into the clinical therapy. So please join me in welcoming Dr. April Pyle.
Thank you so much, Katai. Thank you, Omar. It's a great introduction. Do you see my full screen, or do I need to swap
I would
displays?
say you just have to swap them. We can see
Perfect.
your
There you go.
excellent.
I
Yeah,
never
it
know.
looks good.
Perfect. Okay. Thank you again, Katai, Omar, and everyone. It's my pleasure to tell you about our area of interest. And it's great following Omar because he introduced a lot of the same basic concepts. Um, so, so why are we interested in skeletal muscle? Well, many of you may know, some of you may study skeletal muscle, that there are many different clinical muscle disorders that either result in severe debilitation, lack of ability to use your muscles, or in some cases, death eventually. There's also a lot of mutations associated with developmental defects, muscle aging or sarcopenia, and then genetic diseases, some of which we study in the lab. So skeletal muscle, I would argue, no offense to cardiomyocytes, is one of the coolest organ systems. And that's because actually skeletal muscle is endowed with a muscle stem cell that has the ability to regenerate. And so this is a cell that has the ability to undergo self-renewal, to generate new muscles, new fibers eventually. And this is kind of shown at the bottom here, where you have a cross section of skeletal muscle, and if you have an injury, which we do, all the time, this cell will activate. And this cell that I'm talking about is in green. It's called the muscle stem cell or the satellite cell. And this cell has the ability to essentially give rise to new fibers and recapitulate the entire muscle. And this cell actually does this in concert with a lot of different supportive cells. But the real driver of regeneration is the muscle stem cell. And this cell actually is marked by a transcription factor called PAX7. Many of you know these transcription factors because MyoD was the initial reprogramming factor. If you put MyoD in any cell, this will convert that cell into a myoblast. And so a stem cell, upon activation, turns on a regulatory cascade of transcription factors that, guide you through which type of muscle you have. Myoblasts are the cell that are eventually going to exit the cell cycle and undergo fusion to generate a multinucleated cell. So skeletal muscle is multinucleated, and these myotubes give rise to mature myofibers in vivo. And so in our lab, we're very interested in understanding the process of how to recapitulate the ability of these cells to regenerate and to model diseases using human pluripotent stem cells. So as you heard from Katai, we've spent a lot of time trying to develop disease models in a dish. One that we look at a lot is the disease called Duchenne muscular dystrophy, which is a devastating neuromuscular disease, which is due to mutations in the largest gene in the genome called dystrophin. Mutations in this gene render the skeletal muscle dysfunctional. It sort of acts as a shock absorber in the muscle cell. So if it's not there, the membrane becomes unstable. The complexes at the membrane are lost. This fiber becomes leaky, leaking things like creatine kinase, which is actually what's used to diagnose this disease in the clinic. And so we've been trying to develop therapeutic strategies with iPSCs to model this disease in a dish and to generate therapeutics for patients. And one that I won't talk about, but it's published if you want to learn more about, was published a while back from a graduate student who actually developed a genome editing strategy. And this was done in collaboration with Dr. Melissa Spencer, also at UCLA, where we can actually restore the reading frame of this protein. Um, and and then this will restore dystrophin and functionality to the skeletal muscle cells. And so it's a powerful way to use iPSCs to model diseases, to develop therapeutics, and to think about ways to really have an impact on these patients. This work now, as you heard, is at Myogene, which was at Magnify at UCLA, and now it's in San Diego. And we just had our first pre-IND meeting, which went really well. This is actually using an in vivo AAV-based delivery strategy. But most of what my lab does on the basic end and also building to therapeutic avenues is to generate skeletal muscle in a dish from pluripotent stem cells. So we spent a lot of time trying to optimize this protocol to understand how to generate this mesoderm-derived lineages, which is actually derived from the paraxial mesoderm, not the lateral plate where cardiomyocytes are derived. So we spent a lot of time thinking about that, which I'll show you in the next few slides. And then we also look at the ability of these cells that we make from pluripotent stem cells to be functional muscle, which is critical both in vitro and in vivo. So we look at the potential of these cells to engraft in vivo and make new fibers, which are shown in green, or to have this stem cell function, which I told you in the beginning is so critical for regeneration and skeletal muscle. So we're looking at ways to repopulate muscle in vivo with stem cells generated in a dish. And if there's questions thus far, please stop me. So I can, if not, I'll keep moving. Um, so one of the things that, we're interested in is really how do we not only model diseases in a dish, but to generate skeletal muscle that can be used for cell-based therapies. So what are barriers to using these cells in cell-based therapies? Well, we really want to make sure that what we generate in a dish behaves like in vivo muscle cells. So real muscle, bona fide muscle, skeletal muscle, or, tried and true gold standard skeletal muscle stem cells, as I mentioned, called satellite cells. And we look at the ability of these cells to express the right markers, to have long-term muscle engraftment, as well as ways in which we can really evaluate functionality of these cells in vitro and in vivo. Um, and then, in the lab, we always think about what's the right disease indication and generating preclinical data to really enable scaling eventual safety studies. And this is just a cross section to the left of skeletal muscle, where you can see that we've been able to differentiate pluripotent stem cells into what we call skeletal muscle progenitor cells, or SMPCs. And we do this by transplantation in a dish, and then we isolate the muscle, transplantation in vivo, directly into the muscle, isolate the muscle, section and stain for human myofibers and human cells. So this is kind of our long-term strategy for evaluating skeletal muscle. But how do we generate the skeletal muscle in a dish? So this has actually been quite challenging in the field to date. Cardiomyocytes really have been a default strategy for generating skeletal muscle. And that's because a lot of the first differentiations used BMP, which is well known to activate the lateral plate mesoderm. But as I mentioned, skeletal muscle comes from the paraxial mesoderm, which eventually, so generation through the primitive streak, which is not shown here, And then eventually, skeletal muscle, at least of the limb muscles, is generated through the somite, which is a very intricate and complex three-dimensional structure and development. And so has been challenging to recapitulate until of late in a dish from pluripotent stem cells. And then from there, there are seeds of progenitors or SMPCs that express these markers, as one of which I mentioned, PAX7, which eventually go on to give rise to the fully functioning adult muscle stem cells. And so in the lab, we really strive to understand what are the key growth factors, key signals, key supportive cells that really drive this formation of skeletal muscle, so the paraxial mesoderm at the expense of the lateral plate mesoderm. And so we've spent a lot of time trying to understand how to make somite-like cells in a dish. And next stage are called like dermamyotome-like cells, all of which really closely align with what happens in development. We've studied this in human developmental tissues, age matched to really be able to understand what we're making in a dish. And then we have developed different protocols over the years to try and understand what are the right supportive cells that need to be there in the skeletal muscle differentiations to eventually give rise to the gold standard muscle stem cells or satellite cells. So this is sort of a busy slide, but is an important one because it kind of walks you through our strategies to generate the skeletal muscle in a dish. So as I mentioned, okay, in these current protocols we usually do in 2 dimensions. We can talk about 3D if anybody's interested, but the most of the studies that we've done have been 2D directed differentiation. And so these are the colonies that we start with. And then essentially we dissociate the cells in this process. So really of any directed differentiation strategy, the first 2 to 4 days are really critical. Well, actually starting with even day 0, the quality of your colonies, then the cell numbers that you're using to see the initial differentiations are critical. I'll come back to this. The concentrations of growth factors are critical. And so this process of early specification is really important to generating good skeletal muscle. In fact, if you don't get this process right within the first week, there's really no point in continuing. You really need to go back and start over. And so there's a lot of key markers early on that will really help you determine if you have a good differentiation strategy. And so this is kind of outlined in this cartoon. So essentially we induce mesoderm specification towards a paraxial mesoderm. And one of the key drivers of this, at least in this protocol, is CHIR, which you've already heard about nicely from Omar. So a lot of these growth factors and signaling molecules are also conserved across different protocols, but it is the sort of environment that these cells are in, the media specifications, the concentrations, the cell numbers that really dictate what you're making in a dish. And so in this case, we are differentiating them towards skeletal muscle. You can see we have little differentiation regions. I don't know if you can see it very well, but this is happening when you add CHIR at this time point. These will continue to grow out, and then you will start to see in E6 media, which we use routinely, that you start to see these clusters of cells. And these are quite interesting, which we're evaluating in more detail. But these are really kind of a niche environment that's really helping these initial PAX7 cells to arise. So these are sort of through these first like somite-like clusters in a dish, which you can see. And then eventually we switch to media that supports these PAX7 cells. So things like basic FGF is critical. IGF is important. So we've shown without these that you don't get as good specification of PAX7 cells. And then eventually from these regions where you see these clusters, you will see these muscle cells growing out. This work is published, so you can read more about it. There's also a newer paper that sort of step through some of the specifications that I think is also a nice read. And this is what the skeletal muscle looks like. So you can see we get over 1,000-fold increase in Pac7 in this protocol. And there are robust Pac7 specification as well as fibers. So this is myosins, in this case would probably be myosin heavy chain 3 is a key marker of these differentiated skeletal muscle fibers. And then all around these fibers, you have these Pac7 cells, which really are what we're trying to obtain in these protocols. So you have other markers that tell you in through this process, like 6-1, that's important for understanding like how specific is your differentiations. Again, if you're not seeing these markers, which you can learn more about in these papers, then you probably want to go back and restart and get your cell densities right and your concentrations right, which you can optimize, I'll tell you about. But once we have these Pac7 cells, then we had to identify new markers, which I don't have time to tell you about. It's in this paper here, but this was critical. So all of the standard markers that are published for adult fibers were not working in our differentiation strategy. So we had to identify new cell surface markers to enrich for these cells because the differentiation specification, although there's a lot of Pac7 cells, is not 100% because, as I told you, we have other supportive cells in there that are important for supporting the Pac7 cells and initiating this process. So we're studying how to expand these cells. There's different growth factors. Basic FGF is important. This is still a challenge, a bottleneck, because Pac7, like other key stem cell markers, will go down after enrichment. So there's still a lot of interest in the field on how to keep these cells as high of a stem-like property as possible. But you can make these cells for cell banking, for disease modeling, and we do this routinely in the lab. But as I was mentioning, the first steps are really important in this process. So you really want to understand what you're making in these first, I would say, week or so. And this paper sort of outlines that. So looking across different lines, you can see that it's variable. They're not identical. So these first markers actually are earlier muscle markers, which are called PAX3 and 6-1. And you can use that as a tool to understand how efficient your differentiations are. So this is kind of looking at that. So again, you're using the ROCK inhibitor and then CHIR to really push these cells to paroxysmal mesoderm lineages. And then you have this early mesoderm specification of these skeletal muscle or SMPC progenitors. And if you look across different lines, you can see that their plating density, their differentiation at day 2 is variable. And so what that means is that different HPSC lines have different sensitivities, different survival rates, even initially, and following CHIR addition. And so you have to optimize this per line. And so looking for these double positive 6-1 PAX3 markers is one way to determine how efficient your differentiations are. And then the other critical step is seeding density with CHIR concentrations. And so for every line, you really have to optimize which seeding density should I use per line, and which concentration and for how long should I add CHIR. So you might find that you have the robust markers earlier than, in some lines and others. So it's not a one-shot protocol for every line. You really have to optimize this per line. And so I think if there's anything you get out of this talk, is that every line has variability and you really need to work with that line to see how well my culture is doing, how happy are they surviving upon dissociation. Can I see these early specification steps within this first week? And that's really critical to get a successful skeletal muscle differentiation. But if you do, you can robustly generate fibers, as you can see here. And this is with an RFP reporter line, but you can actually make multinucleated fibers. So this is critical because skeletal muscle, as I mentioned, is multinucleated. So you have multiple nuclei within one myosin positive fiber. And then you have these Pac7 cells, which are the sort of precursors, if you will, to the gold standard muscle stem cells that we're getting, following developmental cues in a dish. Another important feature of skeletal muscle is that you have specific banding patterns in the fibers. So you can look at this in EM. You can look at your myotube formation. You can look at the sarcomeres, which are sort of aligning in these cells in the myotubes that you make in a dish. We've looked at different small molecules that help promote maturation of the fibers, one of which is TGF-beta inhibition or SB. So we use that routinely at the end of the differentiation protocol to sort of enhance the differentiation efficiency. And this is sort of some videos that hopefully you can see of spontaneous twitching, which we will get in a dish if you're really efficiently differentiating these cells. And so this is one line that we're looking at where you can see spontaneous twitching arising from these directed differentiation cultures. This is another view. So, you'll have some regions that are more dense than others. And so maybe you notice these are very different than the cardiomyocyte contractions. And so this is not kind of an undulating, continuous beating, but this is more of a spontaneous induced contraction that you see with skeletal muscle. And so it's very important to sort of understand these distinctions in a dish. But it is really fun when you see them. It's very exciting, and we can do these routinely. And what we really like to do is test the functionality of these cells in vivo. And we like to ask questions about how are they how are they modeling what cells can do, in adult. And so what we look at is how do they engraft in vivo, and how can they repopulate what's called the muscle stem cell niche. And so this is important because, as I mentioned, if you're looking for a cell-based therapy approach, you want your stem cell or your muscle stem cell to be able to get in a niche, which is important for supporting its stemness. And so that's what we sort of look for. This work is published, so I'm not going to spend a lot of time on it, but you can see that we're able to get engraftment. This is sort of what the engraftment looks like. So these green are the fibers that we make. We have regions where there's human mouse. This is an NSG dystrophic mouse. And then we have human-only regions. So we spend a lot of time trying to understand what's happening in vivo during engraftment. And what we learned is that you have populations that are fusing with the in vivo endogenous mouse fiber called chimeric fibers. And we also have human-only fibers. And so we have populations that are making their own human niches. And we've identified markers that dictate what these fibers are. And then we also have chimeric fibers, which are the R cells fusing with the mouse fibers. And so this is something that helps us understand the mature and immature signatures of the cells that we're making, which we look at all the time. And also published, so I'm not going to get into it. You can read about it if you want, or reach out to me separately anytime. I can talk to you about these studies. But essentially, we've been generating mouse studies where we can study niche occupancy of the cells we make from pluripotent stem cells. We know that adult stem cells can make these predominantly chimeric fibers. And now we're embarking on studies that understand how efficient the cells do from pluripotent stem cells. And so now we've been able to show that both developing fibers and also fibers derived from pluripotent stem cells have the ability to get in the niche at low levels and can repopulate after injury, especially in a ablation model where you knock out the endogenous muscle stem cell in the mouse. And so this is sort of what our recent work has shown, that you can get regions of engraftment. You can get pockets of fibers. Here's our Pac7 cells creating their own developing niches, and they're concentrated in regions where there's these human fibers. And so we're excited about this, and we're really working towards moving this to more single muscle disorder cell-based therapy strategies. So there's a lot of people that worked on this project over the years. A lot of the engraftment work was done by Michael Hicks, who now has his own lab at UCI. All of the gene editing work that I didn't talk about in great detail was done by Melissa and Courtney. And a lot of the studies we do is done in collaboration with many people, Catherine, especially for sequencing. And these are the folks in the lab that are now working on different aspects of some of the questions that I mentioned. So I think I'll just stop here so that we can take questions. And sorry, I don't know if I ran over time or not.
No, this was great, April. Thank you. And I should say congratulations on the IND push that you mentioned. I know I'm going to touch maybe a little bit on some therapy stuff. Um, any questions for, I already see one already. Emily has a question. Emily, you want to come off mute?
Oh, sorry. I was just doing an applause emoji.
Ah,
April, thank
okay.
you for your time.
Awesome. I have a quick question,
Yeah.
April, if you don't mind. Just in terms of the fibers that you're specifying, is there a way to look at tensile strength or kind of these fibers, their ability to push, and how do you evaluate that pre and post transplantation? Uh,
Yeah. So this is a great question. So for sure, you can in vitro, you can put them in different sort of stretch systems to look at their functionality. And this is important for disease modeling because that'll really stress the cells and you can start to pull out some of those disease modeling phenotypes. In vivo, in the lab, we have a force transducer system. Sort of, it's called an Aurora system that it actually allows you to put an electrode in the fiber that you transplanted and compared to some ones that you didn't, and look at the force readout or restoration of different force strengths. And so there's a series of assays that you can look at functionally to detect how functional is your muscle that you've transplanted with your cells. And you can do this in the context of putting your cells in a disease model to see like how much fiber restoration is required to restore those functional readouts. And so yeah, it's one of the things that we do. I didn't show in the lab and routinely.
nice.
But great
We have
question.
a Thank you for that. That's a great answer. We have another question from Angel. Angel, do you call me?
Thanks so much. When you did your sorting, I was particularly interested in because cells don't like people being shot through a cannon into a body of like solution. What's your viability and recovery from that? What conditions do you use to improve recovery and make sure you don't have a lot of variability just based on sorting outcomes?
Yeah, this is a great question. And this is something that we still struggle with, just like we struggle with this with the endogenous adult muscle stem cells. So a true adult muscle stem cell in human, or mouse or any model we've looked at is less than 1%. And so really getting that cell, you have to do strenuous sorting. The same is true for our iPSC derived cells. And so what we've found is that actually, if you let the cells, so we've tried different media. So there's no perfect media to support the stemness of the cell yet. So this is something Thermo can work on. But we actually have some medias that will at least keep the cells happy. We've tried different medias, Promocell, Myocult. They have really variability depending on how well your cells are doing. So you might want to try a couple of them out. We sort them. The efficiency is about, depending on the differentiation, the line, anywhere from 5 to 20% of the cultures make these Pac7 cells. So then we sort those out. We are able to expand them as Pac7. But what we found is for engraftment, if we let them sit down for and recover from the sort, that helps the engraftment. If we try and put them in right away, a lot of them will die. They go into this in vivo environment and we don't have the right supportive factors. So letting them recover just for a day or two, not letting them expand necessarily, because then they're going to start to want to fuse. If they get dense, they're going to fuse and make myotubes. That's their job. But if you kind of let them recover for a few days, then engraft them, we found that's the greatest success. But you're right, we still don't have all of the right kind of factors to expand them as true stem cell. And this is an issue in that not just the muscle field, but also the hematopoietic field. And so a lot of these questions are driving kind of future directions we need to work on. Yeah, great question.
What do you sort with really quick?
ERBB3 and NGFR.
No. What system do you use? And then is
Oh.
the expression bimodal, where it's distinctly positive, or is it just a shift, whereas you're doing a high versus low in gating?
Yeah, so we use the Broad Stem Cell Center FACS sorters. We have tried Max, but it's not as high efficiency, but clinically it is better, I guess, for gentler. So we're trying to compare those strategies. But in the lab, routinely we use the FACS based, sorting of the double positive markers, and we have a cocktail of lineage markers that we use to deplete. And it's kind of a high and low. It's not a real large shift. And so I think there's room actually for building in additional negative markers that we need to look at as well. Yeah, great question.
Yeah. Hi, Pyle. These are like
Hi.
very nice. Yeah, very nice presentation.
Thank you.
I have question. Yeah, I have a question about. We know that cardiomyocyte having a spontaneous contraction, but for the muscles to be contracted, have you stimulated those muscles, or it was contracting spontaneously, just like cardiomyocyte?
Great question. So what I didn't say is that alongside, as I said, it's not a pure population, but in our case, that's good because we're co-differentiating neurons in our dish. And so they're becoming innervated, and this is leading to spontaneous contractions. Although you can see with the right stimulations, you can get spontaneous contractions, but we think that having the motor neurons in there are really supporting our fibers. And that's something that we're looking at to really improve that multi-lineage differentiation for skeletal muscle, because once you have that contraction, then that Pax7 cell becomes happier. It's hard to explain, but that's what happens developmentally as well. So we're seeing them arise, but we think a lot of that is due to the support by the motor neurons that are co-developing. And in fact, skeletal muscle in the limb arises from, I didn't get into this, but a neuromesoderm progenitor. So it's a cell that has bipotent potential to make neural and skeletal muscle. And we see both of them co-developing in a dish. And those motor neurons are cross-talking to our skeletal muscle. But that's a whole other
Yeah.
lecture. Yeah.
Yeah. Okay. And Omar was asking about the strength of those muscles. So in line with that, like, can we do a patch clamp to know the strength of the contracting muscles?
Yeah, so people are working on looking at in vitro characterization, just like cardiomyocytes, using like the MEA based readouts as well. So for sure you can do all those functional readouts of the contractions. Yeah, and people are now doing that with disease models. So if you mature them far enough and then you stress them, you can really see some phenotypes now in the field and a lot of Duchenne and other neuromuscular diseases. So we can do similar assays. Yeah.
Okay, great. Thank you.
Mm-hmm.
Yeah, great presentation. So it's pretty similar question, but a little bit different angle in vivo aspect. So for the effective functional integration, transplanted skeletal muscle needed to be incorporated into the local microenvironment, including formation of the neuromuscular junction with the host nervous system. Of course, this one is human versus mouse neuron. It could be a little bit difficult, but to what extent did you observe self-organization and the integration capacity of the transplanted muscle with the surrounding tissue?
This is the question, Katai. You hit it. So this is really where we're headed, is to try and improve and understand that innervation. So we do see some NMJ markers on some of the fibers, but we can't tell right now the extent of the innervation because we're trying to, because what we're seeing in the pockets of the engraftment, we don't know how efficient the mouse are innervating those in the human context. So this is what we're trying to look at now. Also, potentially the need to injure the neural connections because they're happily innervating the mouse fibers currently. So to really look at this. And so I think we need to build in vitro screen for this first, just to understand like what's the innervation cues that are happening in human because they're not the same as mouse necessarily. And so we're trying to look at this, and this is something that one of the students in the lab is looking at. And so we think we're having some low level of innervation, but I think this needs to be improved because innervation, as I mentioned, in vitro is key to maturing the fiber. So once that innervation is happening, then the Pac7 cell is happier. So we don't know fully, but this is something we're looking at. It's a great question, very important.
Great work. Absolutely great work. And super, super exciting to just um I'll steal the ball back, April. Thank you again. This was
Sure.
absolutely amazing. But just to kind of um piggyback a little bit off of what you mentioned in terms of the therapies part, right? We're gonna slightly pivot, but talk about a relevant cell type for therapies too. Um, uh, okay. Can you guys see my slides okay? Excellent. Perfect. And I will say, Tom just texted me. There was, I don't know if folks felt it also in UCLA or in the Southern
Felt
California,
it.
but there was an earthquake.
Yes,
Okay, you
I
felt it?
I was like, okay, should I keep talking? I just kept talking. Sorry, I should have mentioned it.
No, this is good. Tom felt it. He's just going to make sure everything's okay, but we can keep on going on our end. I hope everybody is okay though on you guys' end. If anybody needs to hop off, or we can talk about this again if we need to, but please stay safe. Um, but yeah, the second part of what I wanted to talk about was related to actually what April talked about when it comes to the therapy side of using stem cells. And I put this figure up just as a quick reminder. Don't worry, I'm not going to go through that same spiel I just did. But if you remember, one of the things that we mentioned, right, the mesoderm can form, and sort of April talked about a skeletal muscle being formed from the paraxial mesoderm. And I talked about cardiomyocytes and cardiovascular system being part of the lateral plate mesoderm, right? Cell types that are derived of that, that is. One of the other cell types is blood, right? That's derived from lateral plate mesoderm. And before I talk about this, again, just piggybacking a little bit off of what April was talking about, with pluripotent stem cells, we have the opportunity to use a renewable cell source, right? These cells in culture, if kept happy, as we discussed, can propagate indefinitely. That's part of their what makes them pluripotent stem cells, right? And if we can culture these cells and we can figure out all those intricate pathways that we need to activate or inhibit in order to derive a certain cell type, a terminal cell type, we can actually really start to cement this and use these cells as repair kits, right? Just like what April was talking about with skeletal muscle, that we could potentially transplant back cells into a diseased patient in order to treat different pathologies. Okay? And in this figure, this is a little bit older, right? Not a little bit. This is 20 years old data, but I want to give you just a little bit of an appreciation of how impactful this is, right? If we look at the number of patients affected with various different conditions, right? Cardiovascular, autoimmune, diabetes, cancer, Alzheimer's, Parkinson's, the list goes on. It's a very large number of people that can potentially benefit from having access to a renewable source of tissue that we can use or potentially even engineer in the lab, right, to fix mutations or address certain issues. And in this case, right, stem cells are really, really nicely kind of situated in that pocket where we can use them as a renewable source, and if we figure out how to differentiate them properly, we can get the cell type that we need, right? In order to treat diabetes, I would take my pluripotent stem cells and differentiate them into islet cells, for example. Cardiovascular disease, we just talked about cardiomyocytes. April talked about skeletal muscle and potentially, for example, ALS, right? So there are a bunch of pathologies that we can really address here. And before I get into, and I won't go into too much detail here, but typically there are two different classifications of therapies that we consider, right? The first that you see on the left is called autologous therapies. This is where the cells are taken from a patient, processed somehow, and then reinfused back into that same person. So the cells that are taken out, that are going back into the patient, are of the same genetic material, right? They're the same DNA as the original patient. But there's another method or sort of classification of what's called an allogenic approach or allogenic therapies. This is, if you think about it, more as a universal donor therapy where a donor can donate cells or samples will be collected from a particular donor, processed, and then transfused into a different patient, right? In our case of our workflow with stem cells, right? You can imagine that we can take donor A and collect somatic cells from there, reprogram them into iPSCs, do all the work that we need to do to figure out, how to differentiate them into a particular cell type, and then either infuse them into that same donor for an autologous therapy or a different donor for an allogenic therapy. And there's different considerations for these two types of therapies that I won't get into. I just wanted to kind of put that front of mind first, because one of the cell types that has been making really strong wins in this space are different immune cell types that can be used to target different cancers, right? Most of you will have probably heard of CAR T therapies, where T cells are taken from patients. They're either enriched or modified with a particular cargo, hence the CAR T, right? And then infused back into patients that have particular cancers in order for these cells to target that specific cancer. And T cells have been used a lot in this space, and they've been quite efficacious. But another cell type, immune cell type, that is very, very interesting or could be really interesting here are natural killer cells or NK cells. So NK cells are part of our innate immune system and can actually target pathogens without the need for activation particularly. But if we again think about this in the context of stem cells or iPSCs, we are able to generate iPSCs pretty readily right now. We can use them in both autologous or allogenic workflows, and we have ways in which we can scale them, right? So we can generate, large numbers of these cells when we need them. And for NK cells specifically, right, they are an innately cytotoxic cell. So they can, depending on the certain cues that they receive, they can kill these pathogens or potentially cancerous cells. Unlike T cells, they don't need any HLA matching into donor patients or recipients. And the immune response that's associated with these transfusions, typically, you'll start to see some sort of inflammation or cytokine storm syndrome. They're not as prominent with NK cell therapies, right? So this makes them a great off-the-shelf cancer cell therapy, or particular subtype that we can work towards. And in the field, this has been for the past 2 or 3 years now a really, really hot topic where folks are trying to figure out how we can get functional NK cells in order to potentially use them in this process to treat or target different cancers. And so in a very similar fashion to what we did before, our R&D team developed a protocol where we can take our stem scale spheroids, right? These are the PSCs that are in spheroid form. And then we're going to do the same thing we did before in terms of, mesoderm formation, but you'll notice that this protocol is quite different to what we've seen before, right? So we're going to actually start to specify this lateral plate, this mesoderm into lateral plate mesoderm that will give rise to hematopoietic progenitors, giving rise to hematopoietic stem cells. And then these hematopoietic stem cells, we can then differentiate into natural killer cells specifically. This protocol is around 38 days or so. We start off in our stem scale media, and then we move to our to a media that supports the hematopoietic progenitor population specifically. And you'll notice that during the different stages, we are supplementing the cells with different types of cytokine. So again, very similar to what April was talking about, we really have to make sure that the concentration and the timing of these growth factors that we're adding coincides with when these cells are ready to transition over from the progenitor state into the cell type that we want, right? And I won't go into too much detail again here. I just wanted to show this as another example of another mesoderm lineage that we can differentiate into. One thing I will call out is you'll notice that a lot of the medias here have CTS in front of them, right? So CTS stem scale, CTS stem pro 34, CTS NK expander. Um, CTS is just an abbreviation to what we at Thermo call cell therapy systems. This is a Thermo Fisher terminology for some of our products that are geared towards clinical application, right? So we have a stem scale, our stem scale media, and we have another version of it called CTS stem scale that's manufactured slightly differently. Raw materials could also potentially be slightly different. But the CTS versions of those products, if you see a CTS before a name of a media or a reagent, you'll know that that is part of our cell therapy systems umbrella of products that are geared towards those clinical applications. We have to be really careful with what reagents we use, with what growth factors we use when we are working in settings where the cells that we're working with are going to end up either by the bedside or in clinical manufacturing, right? So just, keep that in mind there. And during this differentiation process, as we follow through, we see the emergence of the right markers that confer those developmental stages that we talked about. So at around day 9, we start to see markers like CD34 and CD90 appear, which are markers of the hematopoietic progenitor populations, right? These are your HSCs that are going to give rise, or NPCs, HPCs that are going to give rise to your immune cell types. But as we specifically differentiate them into NK cells, we start to see that CD56, a marker of NK cells, starts to appear around day 21. And CD16 is an activation marker, which basically a lot of folks look at that to look at whether these cells can carry their cytotoxic activity or can kill. And that starts to also be expressed. But you'll notice that we are not seeing expression of CD3, which is a marker of T cells, right? And this is super important because technically, hematopoietic progenitors can give rise to different immune cell types, T cells, macrophages, monocytes, NK cells, right? And in our protocol, we don't want to specify T cells. We want to specify specifically NK cells. So we have to make sure that we're looking at markers that also exclude populations that we don't want to be in there when we're doing our initial optimization and assessment. And by the end of the protocol, you'll see that the expression of CD56 and CD16 gets even higher, while again, CD3 remains negative. So we're specifying specifically natural killer cells. And I won't get into too much detail again here, but the idea is that we put these cells into through an assessment of a panel of different markers, cell surface markers, that a lot of folks commonly look at when they're trying to see if these cells can actively carry out their cytotoxic function, right? Whether they can kill cells or not. And we see, the expression of these activation and inhibition markers post-enrichment in our NK cell populations that we've specified. But more importantly is an assessment of that function itself, right? Not just looking at markers, but we want to see if you were to put, a cancer cell next to this INK cell that we've specified, is it going to carry out its function and potentially kill that cancerous cell or the pathogen? And so to answer this, we actually utilized a completely different system that we have. We have our media culture system called our oncoprotumoroid culture media. This has nothing to do with stem cells. This media, basically what it allows you to do is to take primary cancer tissue from patients, mainly epithelial in nature, and culture them as 3D tumoroids, right? Or 3D tumor organoids. And we're able to do this with a human colorectal cell line that we labeled with GFP. And we co-cultured it with our NK cells that we've specified in the protocol that I just showed you. Right? So we're doing a co-culture here where we have colorectal cancer cells that are GFP positive, and then we're adding in at time 0, as you see here, at different concentrations, we're adding in natural killer cells. And you'll see that as soon as we add those natural killer cells in, the GFP signal, which is indicative of the presence of these cancer cells, drops dramatically in the first 24 to 48 hours. And it actually does so in a dose-dependent manner. So the more NK cells that we add, the faster we see the GFP and subsequently the cancer cells be eliminated. And we can confirm that we also see an increase in caspase 3-7 activity, which is a marker of the apoptotic pathway. Again, confirming that these cells are being killed by these NK cells that we're adding in culture, which is really, really cool, right? Super, super promising that we have a proof of concept here where we can, in a 3D format, generate these very large number of these NK cells pretty quickly, pretty efficiently. And if we need to, we can use them in certain cases to eliminate certain cancer cell types in the future. So just wanted to share some of this work with you, but I'll end there. I know this lecture was a little bit longer than our usual, not by long though, and it's usually my fault. I talk too much, but I hope you really appreciate how really, really cool this is in terms of a system from start to finish of growing stem cells and then differentiating them into these really, really cool cell types that we can, we have use for in the clinic as well. And with that, let's see if we have any questions in the, where should I see? Any questions in the chat? No questions in the chat so far. Any questions anybody had or?
Hi, Omar. Again, I have one question. Can I ask?
Yeah, please go ahead.
So yeah, about the NK cells that you have shown, it's like very interesting and new to me. So yeah, actually, if we compare the culture that we do using the cardiomyocyte, iPSC and the cardiomyocyte, so if we compare the workload and the ease of the work, like how easy is doing like working on the cardiomyocyte and the working on the NK cell differentiation and all that. Is that in the
Yeah,
preliminary stage or like we can handle those cells quite easily as we do for the cardiomyocyte?
it could be a little bit tricky. So on the cardiomyocyte side, they could be a little bit fragile, especially if they get a little bit bigger. On the NK protocol, because the protocol is a little bit longer too, Naresh, it could be a little bit more challenging just because you have to make sure you're specifying the right progenitor states, right? And again, I'll reference what April said at the beginning of her talk. It's really important to evaluate the different stages throughout your differentiation protocol. There is no need to go all the way to the end of day, 38 if you know that by day, 4 or 5 that the markers that should be there are not. right? So to that point, just because that protocol is a little bit longer on the NK side, it could be a little bit more challenging there. But um, we see potentially slightly more cell types that we can identify at the moment with the 3D cardiomyocyte protocol. So again, making that a little bit more complex. So it's really apples and oranges too, Naresh. It's hard to compare. But don't worry, we are here to
Yeah.
help, right?
Okay.
I will say this every time, and uh, please, use your resources. As a field, we've come a very, very long way, and it wasn't by accident. We have a strong sense of collaboration within the stem cell community. Um, uh, we have a lot of know-how and a lot of expertise that we can refer back to. There are, I mentioned the cores at your institutions, the researchers that you're working with, the vendors that you could potentially be working with, right? We all have different resources, so feel free to reach out to any and all people who you think will be helpful. And I'm sure you'll find somebody to help you along. And on that front, I'm definitely happy to help along. So let me know what you need. And if you need to start one protocol, if we need to go through both of them to see what you
Okay.
would prefer to work with or what sounds more interesting or potentially an easier lift for a project here or project there, let us know. We're happy to help.
Yeah. Another question. So
Yeah.
you introduced another term like your product is for stem cell therapy, something.
Mhm.
Uh, yeah. So
Cell
in
therapy
context of
systems. Yeah.
cell therapy system. So normally, like for medicinal use, or the product that we are getting, those are only it says that only for research purpose. So do you
Mhm.
think those products can be used for the medicinal use,
So
for the medical use? I
it's
mean.
not, yeah, it's not exclusionary. You can, but you'll notice that for clinical manufacturing, there are certain guidelines when it comes to and regulations when it comes to the type of material and the source of that material that you can use. And this is really intended to as a safety measure, a precautionary measure. So, for example, what we try to do with the RUO or the research use only products is we try to remove animal origin components, right? That could potentially introduce different viruses or pathogens. We try to replace these with recombinant proteins or human derived alternates so that you're not having to go to an animal source for that because these potentially final products will be used in a clinic, right? That's not to say that you can't use things like, for example, FBS that comes from animals we use routinely in culture for various different applications. That's not to say that you cannot use this for your IND applications with the regulatory bodies like the FDA, but it's um, a, a much harder justification as to why you would want to use this particular reagent. With the CTS offerings, we try to eliminate some of that for you in the get-go by making products that don't have these components that are also manufactured in a very strict setting that is compliant with clinical manufacturing for bedside therapies, patients, things like that, and make that transition a little bit easier.
Okay.
Great question.
Thanks so much. Thanks.
Yep, of course. Awesome. Any more questions before we call this a day? And again, I really appreciate all your questions and your engagement. This has been super, super rewarding for me and Tom. I can speak on our ends. Um, I think Kitai next week is the final lecture, if I'm not mistaken.
Yes,
Um,
next week is anyhow today, Omar, and there was April, one of the really good lectures we experienced, and then also really good discussion we did. I really appreciate. And the next week is we are talking about the neural differentiation, and then also another best leading scientist, Michael Well, will present his work in brain study. So therefore, this is also someone who study, oh, no, I'm not working in brain. No, that is not true. Actually, it is very useful, even you are working on the other tissue type. And then brain is also ectodermal lineage. So therefore, any skin, hair, tooth, or whatever. And of course, brain is one of the key hallmark in the regenerative medicine. And then also when we study how brain ectodermal lineage is differentiate and how difference between mesodome and the endodome, that is really, really useful information when you do the troubleshooting of your cell differentiation. So therefore, even if this is not your tissue type interest, always is beneficial. I really appreciate.
Excellent. Thank you. And thank you, April. That was a great talk as well. I appreciate you joining us today as well.
Thank you, everyone.
Perfect. Thanks, everybody. Have a great rest of your week.
Thanks, all.